So, dear students, good afternoon. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute. Today is 10th September 2022. I'm very happy to be in this class, and today we are going to discuss movement of nutrients in soil and their uptake by plant roots. So this is really very important topic, and I am teaching this subject for the first time to PG students. I tried to collect information from different sources and try to make the meaning out of that, because it is really very complicated subject. It is not very easy to understand. So I tried to make it, I tried to simplify this subject as far as possible so that we can understand. And there may be some errors, some mistakes here and there. So if you come across any controversy, any mistake, any problem with what I teach or what I say, kindly point it out so that I can correct myself. It is likely that there may be some errors in this presentation, in this talk. So because I did my best to get the correct information, but there are so many theories, so many views, so many confusion. It is very elaborative subject. So therefore, just to get some idea, some glimpse, we can you can see this topic. You can deal with this topic. Learning objectives are to learn the mechanism of movement of ions to plant roots. So first part is that we will see how the nutrients are reaching the plant roots so that they can be taken up by the plants. Or is it the root which is reaching to the nutrients? So first thing is that plants should come close to the roots. Second is describe the process of nutrient uptake by plant. So nutrient uptake itself is not completed. Nutrient uptake means entry of nutrient into plant. Entry of nutrient into plant roots is your nutrient uptake with respect to nutrient uptake by root. But it also includes the movement or transport of nutrients within the root system, how it moves within the root system so that it can be assimilated by plants, or it can be used by different parts of the plant, or it can reach to the leaf. Because it is starting from root, it has to first enter the root, and through root, it has to move into the plant system. It is required to be distributed in the plant system. And as you know, that nutrient and water both move in the plant system through xylem. So main thing is that these nutrient move through xylem, water move through xylem, and carbohydrates or food synthesized by leaves move in phloem. Through phloem, it passes to different parts of the plant, including fruit, flower, your stem, and then your roots. So these are both are your vascular bundles. I think you know about vascular bundles, xylem and phloem. So we learn some fundamental things, then move to some advanced aspect of this. Let us see, sometimes it is active transport. Mostly people say that nutrient uptake is done by two processes, active transport and passive transport. And later on, you will know there is one more kind of transport is also there, that is membrane transport. So active transport of nutrient means movement of molecules, ions across a membrane from a region of lower concentration to a region of higher concentration. This is important. So this may include your molecules like boric acid. It may include certain other molecules, organic molecules, or it may be ions. So movement of molecules or ion across a membrane. Membrane is basically your plasma membrane. If you know the plant cell structure, outside you have cell wall, then you have cell membrane. So that is also called as plasma lemma. Plasma lemma is another name for cell membrane. So it has to enter and pass through the cell membrane. Cell membrane is selective. It selectively allows certain chemicals, certain molecules, certain ions to get in and certain to get out. So it controls the movement of ions and molecules. So active transport means it has to pass through the membrane from region of lower concentration to region of higher concentration. Because outside the plant plant roots, a substance, an ion and a molecule may be in lesser concentration and it may be more inside the plant. So therefore, it is against the concentration gradient. So therefore, you require some energy because you are against the concentration gradient. See, if you have a boat and moving on water, if it is on the direction of the water, you are moving the boat, then it is fine. And if you are moving your boat against the flow of the river, then you have to put lot of energy. So similarly, it is against the wind or against the gradient. Therefore, you need energy. Apoplastic movement, you will come across, you will know more about apoplastic movement. Apoplast is the space outside the plasma membrane. So it is outside the plasma membrane within which material can diffuse freely. Means in this space, apoplastic space, you will see it, the materials, here material means ions, molecules, can diffuse freely. It is slowed down by the Casparian strip in roots. So actually what is happening, the first thing is that it has to pass through the membrane and then it can diffuse freely. Diffusion means again from higher concentration to lower concentration. It is slowed down by the Casparian strip in roots. So later on, if you have seen the structure of the roots, you will find some bands or some layers of Casparian strips in the root systems. So they are these Casparian strips stops or reduces the movement of ions or movement of material. It allows only selected materials to go in and it does not allow some materials to come out. So Casparian strips play important role in the movement of ions. Then there are certain nutrient carriers which take the nutrients from one place to another place, one cell to another cell. So these are nutrient carriers. So some people explain the transport of nutrient within the plant cells or within the roots through some nutrient carriers. A theory explaining the transport of nutrients from the surface of root into the root cells, occurring by means of carriers. So these carriers may be materials actually, organic materials, proteins, enzymes, something like that, that carries the nutrient. Next, plasma membrane or plasma lemma. It is microscopic membrane made up of lipids and proteins. So here I suggest you to study the structure of plasma lemma or plasma membrane. It encloses the protoplasm. You know, you might be knowing it encloses the protoplasm. So you should know the structure of the plasma membrane. This is very, very important for a student for agronomy also, to know the structure of the plasma membrane. From any standard textbook of plant physiology, you can find a structure of plasma membrane. So it forms external boundary of the cytoplasm. It is cytoplasm boundary. It is boundary of the cytoplasm of a cell or encloses a vacuole. So plasma membrane is there at the cell level as well as vacuole. This vacuole also have some membrane. Plasma membrane kind of membrane is there. So vacuole is also important in the transport of nutrient. Sometimes it stores the nutrient and then nutrients passes through the vacuole in the cell. So therefore it is quite complicated. The nutrients need to move from one cell to another cell, and these cells are of different types. If we start from the outside the root, we start from the epidermis. The characteristics of epidermis cell will be different from the next cortex cells, pericycle, and then xylem, phloem, Casparian. So these cells, they are of course cells, they contain vacuole also. Every cell will have vacuoles. So sometimes a nutrient has to pass from cell to cell via vacuole. First, it will enter vacuole, then it will come out from vacuole, go to the next cell by plasmodesma, and then and again the same process. And sometimes it may escape the vacuole and directly from cytoplasm of one cell, it will move to the cytoplasm of another cell. So there can be different routes of transport of nutrients through the cells. Symplastic movement. Symplastic movement means the diffusion or movement of water or low molecular weight solutes. So these solutes are molecules or kind of ions in the inner side of the plasma membrane. Inner side is your inside the cell, main part of the cell, is called as symplastic movement. So movement from cell to cell is your symplastic movement. Now, nutrient supplying power of the soil. The capacity of the soil to supply nutrients to growing plants from the labile, exchangeable, and moderately available forms. Diffusion coefficient, today we will see in detail. Proportionality constant that indicates the ability of material to allow gases and ions to flow under a partial pressure or concentration gradient. So that is your diffusion coefficient. We should know the meaning of this. Nutrient uptake, process of ingesting nutrients. Ingesting means taking in by plant roots, or it is the transfer of the nutrient ions across the soil root interfaces into the plant cell. So here nutrient uptake got narrow meaning. It is just entry of nutrients into the plant. However, nutrient uptake will be completed only when this nutrient is reaching the xylem cells. And in xylem cell, it will easily pass to different parts once it is in xylem cell. So it has to move, it has to run from cell wall up to the xylem. It has to reach up to the xylem, and in reaching there, it is transport. It is transported from cell wall up to the xylem. So you can say uptake of the nutrient will be considered completed when it is there in the xylem. So normally they write the term nutrient uptake and transport. Nutrient uptake and transport. But if I say nutrient uptake, it involves reaching nutrient up to xylem. So that may be acquisition or acquiring the nutrient or uptake of the nutrient. Labile, a substance that is readily transformed or used by microbes easily. Nutrient concentration content, I told you already. Nutrient efficient plant, a plant that absorbs, translocates, or utilizes more of a specific nutrient than another plant under conditions of relatively low nutrient availability in the soil. Suppose iron is deficient in a particular soil, and you have sown 10 different varieties of a crop, and say 8 varieties are showing deficiency symptoms. They are susceptible, but 2 varieties are not showing deficiency. That means those 2 varieties are iron use efficient varieties or iron use efficient genotype. Or in simpler terms, you can say nutrient use efficient genotype. Means under low supply of nutrients, they are able to grow successfully. They do not have any limitation of that particular nutrient. So therefore, and they are nutrient specific. It is not that one genotype which is efficient for iron may be efficient for zinc. It is normally not happening. Or mostly they are nutrient specific. For one nutrient, you will have one particular genotype. One genotype may not be nutrient use efficiency for 2 or 3 or 4 nutrients. Normally it is for one nutrient. So nutrient use efficient plant, genotype or variety. Now question comes how it becomes nutrient use efficient and how others are not. Mainly because of the extensive root system, the efficient root system, and it has got certain siderophores or phytosiderophores that can solubilize or that can improve the losses of that nutrient. They can keep it in the soil. They can mobilize that nutrient to the roots, and the plant is able to utilize more efficiently. Like we know, more crop per drop, more crop per drop. So more biomass, more yield per unit of that nutrient absorbed. So plant have two mechanisms. One is uptake, other is utilization. So in uptake, it is efficient. It is removing whatever is available to the best, and then it is utilizing it to the best. So therefore, it is nutrient use efficient plant. Now, first thing is that how nutrients are reaching the roots. That is important part. And all of you know about it, that movement of nutrients from soil to roots. So nutrients move by three mechanisms to plant roots. One is root interception. Means root is going to the nutrient. Root will intercept the nutrient. It will pass through the soil solution. Root will go up to the cation exchangeable site also. Certain portion of root or rootlets will touch the nutrients directly. They will come in the contact of the nutrient, which is normally called as root interception. Means root is reaching the nutrient. Second one is mass flow and diffusion. In this, the nutrients are coming to the root. So you can see here, mass flow on the left side of this picture. They are in a higher concentration, and along the water, they are moving to the roots, or moving to the, you can say, soil solution. Moving to the soil solution in mass flow, or from soil solution, roots can easily take up these nutrients. So first thing is that nutrient from different parts of the soil should go to the soil solution around the root. Soil solution may be there throughout the soil system, but whatever soil system is near the root, in that it will have soil solution, and these nutrients should reach there through mass flow with the help of water from higher concentration to lower concentration. So this is plant, and somewhere here is your nutrient. Soil solution is here. Soil solution is here. And then, I'm sorry. They are moving from different sites and soil solution to the roots. They are coming near the root through the mass flow. So through mass flow, they are reaching the roots. They are moving from soil solution and other sources like exchangeable sources, they will exchange with the soil solution. The nutrient will come into the soil solution from exchangeable side. And once they are in mass, they are accumulated here, they will start flowing towards the root. So mass flow is from soil solution to the roots. Similarly, diffusion. From soil solution, they are in higher concentration, and then around the root. Because around the root, the nutrients have been taken up, so their levels are low here. The nutrient concentration is less here. Here it is more in soil solution. So from higher concentration to lower concentration. And why there is lower concentration here? Because roots are continuously removing the nutrient. So in diffusion, they are moving from higher concentration to lower concentration. And root interception or is directly, roots are in the contact with the soil solution. They are in the direct contact of the soil solution. So hope you are able to understand these three things: mass flow, diffusion, and root interception. Any doubt, any problem in this? Now we study the root interception, how it happens, how plant will take up nutrient directly in the solution. So exchange of ions through physical contacts. First, they will come in the contact with each other. Exchange of ions through physical contacts between root and mineral surface, enhanced by increased root biomass. So the root interception will be more if you have more root biomass and proliferation, its distribution in the larger volume of the soil. And then what happens? Roots extend to soil solution. Soil surfaces, even they can go to exchangeable site also to access the nutrients. So nutrients which are there in the soil solution, root will be there, but it can reach to the exchange site also, wherever possible. And it will extract the nutrient from there. And then what is mechanism there? Mechanism there, when the root and the soil solution or nutrient come in the direct contact. So it is by oscillation. Oscillation, you will see. So hydrogen ion adsorb on the surface of the root hairs. So you know when exchange happens, the hydrogen ions come out from the cells and their concentration is more around the root. So outside the root, you get hydrogen ion adsorbed. They may be on the root surface or on the root hairs and exchange with the ions held on the clay or organic matter surface or in the soil solution. So wherever nutrients are there, they can be exchanged directly with the hydrogen ion in root interception or root context, root nutrient contact, and by oscillation, by shaking. So ions held by electrostatic forces at these sites oscillate within a certain volume. So ions which are there on the roots, they are hydrogen ion, and other ion which is in soil solution or exchangeable site will also oscillate, or kind of dancing will happen. Both will dance, attract each other and exchange with each other. So when the oscillation volume of two ions, H ion and K ion overlap, when they touch each other while dancing, they are exchanged. So ion exchange occurs by dancing, I can say dancing. Calcium ion on clay surface could be absorbed by root. Calcium can be replaced, potassium can be replaced, but this is mostly positively charged ions. So you can see conceptual model for root interception or contact exchange of nutrients between ions on soil or in soil solution or in root exchange sites. Overlapping oscillation volumes cause exchange of hydrogen ion on the root with the potassium ion on the clay mineral surface. So this is just shown you wherever the nutrient is, whether it is on the clay, whether it is on the humus, whether it is in the soil solution, it will oscillate, it will start dancing or it get attracted towards the root. And here you got hydrogen, it will also oscillate, and the region, the circling area will increase. And once they dance or they oscillate, they come close contact and they change. One will come to the left, other will go to the right or to the root. So this is very interesting, root interception, and hope you could follow it. And now a question comes that there are three mechanisms. One is mass flow, other is diffusion, and third one is contact exchange. So most or many nutrients are coming to the plant root through mass flow and diffusion. And contact interception is not very common. It is only in case of calcium where it is important, root interception. So let us see which mechanism is more important for which nutrient for their transport to the root. So significance of root interception, mass flow, and diffusion in ion transport to corn plant. This is from Havlin book, you can also see. So it is just in percentage. Suppose for nitrogen, just you don't bother about this column. See the interception, root interception, it is just 1%. So nitrogen is transported to the root, 99% is transported to the root by mass flow and 0 by diffusion. So this is important for nitrogen. Coming to phosphorus, almost 94% is transported by diffusion. For phosphorus, it is mostly diffusion, and only 4% by mass flow and 2% by root interception. Coming to potassium, so again, like phosphorus, 78% of potassium is transported by diffusion and only 20% by mass flow and 2% by root interception. See the case of calcium. So you see percentage supplied by, it is just percentage supplied by. So here, if you make the total, it is 100. Here it is 100 up to NPK. But in case of calcium, you can read these three lines to understand. if it is not in percentage. So you can see if the diffusion value are 0, if the diffusion value are 0, these are relative values actually. These are relative value. So 440 means majority of the calcium will be taken by mass flow and then by partly by root interception. Similarly, next for magnesium, it is 0 value. Here it is 280, means you can see just the ratio of mass flow and root interception. See these two ratios and then convert to percentage. So here also you can conclude that most of the calcium and magnesium also move through mass flow. Similarly, copper also move through mass flow and boron also move through mass flow. So kindly study this table and try to understand how these nutrients are transported to the roots or how they come in the contact of the roots. It continues. Now, very interesting fact that many a times our plants, our field crops are infected by mycorrhiza. So mycorrhiza are important. There are two kinds of mycorrhiza. One is ectomycorrhiza, other is endomycorrhiza. So ectomycorrhiza is mostly on the outside surface of the roots and they make a small entry into the plant cells, interspaces. And they are mostly found in trees, in temperate areas. And they are not very common in tropical areas and subtropical areas. In tropical and subtropical areas, these endomycorrhiza are very common. But now people don't call them by the name endomycorrhiza. A new name has been given to endomycorrhiza, which is AM fungi, arbuscular mycorrhiza fungi. So now you have two kinds of fungi. One is ectomycorrhiza, other is arbuscular mycorrhiza, AM fungi. AM fungi, AMF. Sometimes people used to call them vesicle, VAM, vesicular arbuscular mycorrhiza. Now that name is also obsolete. We should not call endomycorrhiza as VAM. They should be called as AM fungi, arbuscular mycorrhiza fungi, because a vesicular mycorrhiza, vesicular arbuscular mycorrhiza VAM, all were not making the vesicles always. They were not all the mycorrhizal fungi, endophytic mycorrhizal fungi, they were not making the vesicles always. Vesicle is a structure of fungi or fungus. So then their name was changed to AM fungi, arbuscular mycorrhiza fungi. So what happens? Most of their part enters the plant cells and through intercellular spaces, they can enter into the plant cell. So they are actually intracellular. They enter the cell itself. They enter into the cell parts and there they make certain bodies. And those bodies are arbuscules. Those bodies are called as arbuscule, which are exchange site. So their exchange happens. Plant or mycorrhiza would like to absorb or would like to take carbohydrate for its life. And in return, it will give some nutrients to the plant cell. What are the nutrients? It can give phosphorus, it can give zinc, and it can also give some other nutrients. But in most of the cases, it supplies phosphorus and zinc. So therefore, they are also helping in the uptake of the nutrients. So that is why we cannot separate them from the roots. They are doing the function of the roots. So therefore, studies of mycorrhiza in nutrient uptake is also important, like root uptake. So we can treat it also as a kind of root uptake, or they can be called as important for uptake of the nutrients. So root interception may be enhanced by mycorrhiza, more benefits to plant in infertile cells. Where you get less fertile soils, there you will find more use, because they can release some chemicals into the soil which can solubilize the phosphorus and increase the mobilization or availability of phosphorus. Then they can increase the root absorptive area. The absorption area is increased, and also importantly, they are finer than the root hairs. They are finer than the root hairs, so they can enter the plant or they can enter the soil where your root hairs cannot enter. So therefore, the larger volume of soil can be explored in addition to roots by mycorrhiza. So they are more effective in infertile or less fertile soils where phosphorus is less in the soil. Under phosphorus stressed environment, mycorrhiza are more important. So mycorrhizal colonization is enhanced by slightly acidic conditions. As you know that mycorrhiza or fungi like to grow more in acidic soils. Low phosphorus soils, they love to grow in low phosphorus soils. Adequate nitrogen is required and low soil temperature is required for their best growth. The hyphal threads act as an extension of plant root system. So they are helping the plant root system in the acquisition of nutrients. So therefore, greater soil content. So you can see here in this picture. So these are mycorrhiza. You can see that they are increasing the absorption area for water, for nutrients. Now, I think I have told, two groups of mycorrhiza are there, ectomycorrhiza, endomycorrhiza. Ectomycorrhiza are on the tree species of temperate zones, also in semi-arid zones. Endomycorrhiza more widespread, and they are in tropical and subtropical area. So endomycorrhiza are your AM fungi, arbuscular mycorrhiza fungi. So roots of most agronomic crops are infected by AM fungi, and it grows into the cortex. You know, cortex is the part of the roots. Inside the root is structure. If you get transverse section of root, TS section of the root, you will find these cortex cells. Inside the plant cells, a small structure, arbuscules are also there, which are inside the cell of the roots. The transfer of nutrients from fungi to host plant happens. AM fungi, larger nutrient absorption area by AM fungi can be up to 10 times that of uninfected roots. So larger area for acquisition and uptake of nutrients. Fungal hyphae can extend up to 8 centimeters into the soil, thus increasing the absorption of nutrients. Enhanced phosphorus uptake is primary cause of the improved plant growth due to infection of mycorrhiza. Now you can see very important slide, effect of inoculation of endomycorrhiza, your AM fungi, and phosphorus on nutrient content in corn shoots. The nutrient concentration in corn shoots in microgram. You can see there are two cases. No phosphorus is applied to the soil, and 25 ppm phosphorus is applied to the soil. 25 ppm means around 50 kg P per hectare was applied. 25 ppm roughly means 50 kg. You multiply by 2, that will give you kg per hectare. Or you can multiply by 2.24 exactly to get phosphorus applied. So 25 ppm P was added. There are two cases, without P and with P. So there are two cases here. No mycorrhiza was inoculated from outside. And here mycorrhiza was inoculated in the soil. So what happens? Sometimes mycorrhiza are there in the soil, but they are not sufficient to fully help the plant. But if we increase their population by adding them from outside or inoculating them into the soil by seed or by some other method, and artificial inoculation will increase their number in the soil. So here are two cases, no mycorrhiza was added, and here mycorrhiza was added. So you can see the difference. When no phosphorus is applied, here the concentration was less, here the concentration was more. So you can see the difference, 6,000, 9,700 a microgram, 1,200. So for all the nutrients, you can see what is happening. Now, even in this case also, no mycorrhiza. Here phosphorus was added, but there was no mycorrhiza. Then the uptake of phosphorus addition on different nutrients. So you can see 2,970, 70. And here also, So what you can conclude from this slide is that mycorrhiza inoculation is useful. Number two, mycorrhiza inoculation improves the phosphorus uptake and it enhances the concentration of phosphorus and other nutrients. Even potassium, you can see potassium concentration improved under both. So you can see it is not just phosphorus or not just zinc. Mycorrhiza help in increasing the uptake of other nutrients also. You can remember, though the contribution may be less, here also you can see contribution is less of mycorrhiza inoculation, but mycorrhiza are helping to increase the uptake of other nutrients also. So most of the time students are asked by a question. So many scientists, many people consider that it is only phosphorus uptake which is affected by mycorrhiza. But this data shows you, it is before you, that mycorrhiza help in the increasing the concentration and uptake of not just phosphorus or zinc. It increases uptake and concentration of potassium, calcium, magnesium, and other nutrients also. It will not discriminate. It will take up everything available in the soil. So we will go to the last link and then finish. I am going very slow in this lecture. So it may take 3-4 lectures to complete this topic. I want to go slowly in this so that we can understand in detail these concepts. Thank you. So we go to the next link. Hope you could follow the meaning of role of mycorrhiza in nutrient uptake. And now we go to mass flow. So ions of soil solution are transported to roots. So they are coming from soil solution in mass flow to root as a result of water uptake, transpiration. So transpiration is there. So water also moves from soil solution. Solution means they are in water. They are in water. So with the water, they move to the roots. And because of evaporation and transpiration, this water moves. And percolation in soil. So ions of soil solution are transported to root as a result of water uptake, that is transpiration by plant, evaporation, soil surface, and percolation of water in soil profile. Important factor in supplying nutrients to plant, mass flow. The quantity of nutrients reaching roots is determined by rate of water flow or water consumption and average nutrient concentration in soil water. So this is important. So therefore, nutrient and water have very positive interaction, very, very positive interaction. I told you sometimes back also, if you need to have sufficient water in the soil to make the nutrients available to the plant. If there is no sufficient water, the nutrient will not solubilize, they will not be part of soil solution, number one. Number two, even their movement through mass flow will not be possible if soil is not having water. So therefore, nutrient water interaction is very, very important, and I wish to take one lecture on this topic also. At later stage, of course, not now. If you would agree, then only I will take a lecture on nutrient water interaction, which is a difficult subject to understand. So it is important factor in supplying nutrients to plants. The quantity of nutrients reaching roots is determined by rate of water flow or water consumption and average nutrient concentration in soil water. These two things affect the mass flow. Now, can supply an excess of calcium, magnesium, some micronutrients, and most soluble nutrients. So it can supply, means mass flow, through mass flow, it can supply calcium, magnesium. You have already seen some micronutrients and most soluble nutrients like nitrate, chloride, and sulfate. As soil moisture is reduced, water transport to the root surface decreases. So if water transport to the root surface decreases, means mass flow will decrease. So reduced at low temperature, also temperature will affect the mass flow. So reduced at low temperature because of reduced transpiration and reduced evaporation. Now we go to the diffusion. As you know, the meaning of diffusion, ion moves from an area of high concentration to one of low concentration. Roots absorb nutrients from surrounding soil. Roots absorb nutrients from surrounding soil solution. The nutrient concentration at roots surface decreases compared with the bulk soil solution concentration. So nutrient concentration gradient is established and most of the P and K move to the root by diffusion. So from higher concentration, which is there in the solution, and they move to the root surface. Now, can I ask a difference in mass flow and diffusion? Any student can tell me, can try, at least you can try now. What is the main difference in mass flow and diffusion of nutrients? Please try. Subhranshu, can you try? What is a difference in mass flow and diffusion?
Yes, sir.
Yeah, what is difference? Any idea? Anybody? Are you there or not?
Yes, sir.
Everybody is there? Okay.
Sir, in diffusion, the concentration difference is there, but in mass flow, there is no role of concentration and their difference, I think.
Yeah, you are right. You are right. In mass flow, of course, constant gradient is there in that case also, because in mass flow also, I have shown you that there is concentration gradient. Just let me show you some more. Ions of soil solution are transported to root as a result of water uptake, transpiration by plant, evaporation soil, and percolation of water. Yeah, I think the quantity of nutrient reaching root is determined by, yeah, I think you are right. Because in this subject is really new to me. So I'm also learning, I'm reading, I read some books on this topic, and that could be right, what you tell. Because here there is no uh, concentration. So here they will simply move, whether their concentration is high or low, they will move with the water. You are right. And in case of diffusion, they move from higher concentration to lower concentration. I think it should be okay. What others feel? It is mainly driven by moisture. It is, if water is moving, they will move, irrespective of the concentration. Is that correct?
Yes, sir. Also, the mass flow is also influenced by the transpiration pool as well.
Okay, and it will not remain unaffected or less affected. Anyway, this is just, you can have some interest in this so that at least you can try, you can read more. Now we see move on to diffusion. Ions or molecules move from area of high concentration to low concentration. Roots absorb nutrients from surrounding soil. So nearby nutrients are removed and lower concentration will be there. And in solution or in bulk soil, you have higher concentration. So ions will move with the concentration gradient, and most of the phosphorus and potassium move to root by diffusion, and nitrogen by mass flow. Now, diffusion occurs based on Fick's law, which is given by the equation F is equal to minus D. This means it is what are the factors that will affect the diffusion. So minus D dC upon dx, whereas F is diffusion rate of diffusion or quantity diffused per unit cross section or per unit time. dx is concentration gradient. What is the difference in concentration gradient? C is the concentration, and D is diffusion gradient, and X is the distance. So you can understand this formula means these factors will affect the speed of diffusion. What will affect the speed of diffusion? Concentration gradient, concentration, diffusion gradient, and distance. So these are internal factors. They will affect the diffusion rate. You can go into the details of this kind of formula if you have interest. So ion uptake by roots or create or maintain diffusion gradient is strongly influenced by temperature. So besides those factors, other external factors are temperature affecting diffusion. So within the range of about 10 to 30 degrees centigrade, an increase of 10 degrees centigrade usually causes the rate of ion absorption to go up by a factor of 2 or more. Means from 10 to 30, this is applicable only from 10 to 30. So if you go from 10 to 20 or 20 to 30, then this apply, and beyond 30 it will not apply. So absorption to go up by a factor of 2 or more. Slow under most soil conditions and occur over very short distances. So it is possible only in short distances. Typical diffusion distances are 1 centimeter for nitrogen. Students must remember these values. They can come to your quizzes and some exams. How much nitrogen can move by diffusion? Answer is 1 centimeter. How much K potassium can move by diffusion? 0.2 centimeter. And how much P can move by diffusion? It can move by 0.02 centimeter. So you can see the differences. Of course, nitrogen is primarily moving by mass flow, but if you compare its diffusion also, then it is 1 centimeter. And K and P is the least. So P is the laziest element, which is not even liking your diffusion. It moves very slower distance. So therefore, you apply this phosphatic fertilizer near the roots. Roots do not absorb all nutrients at same rate, causing certain ions to accumulate at root surface. Thus, it results in back diffusion. So sometimes back diffusion can also happen if concentration of ions or nutrient is more on the roots or near the roots, where the concentration gradient is away from root surface and back towards the bulk soil. Nutrient diffusion away from root is much less common than diffusion towards the root. Okay, but it can also happen. Or you can say reverse diffusion can also happen. Importance of diffusion and mass flow in supplying ions to root surface depends on ability of solid phase of soil to replenish or buffer the soil solution. So solid phase is your whether colloids, it is mostly colloid, how much they can replenish the solution or how much they can buffer the soil solution. So that is also important. If there is no source in the solution, there are no nutrients in the solution. So then what it will transport? So buffering capacity of nutrients should be high. Ion concentration influenced by the type of clay, minerals in the soil, and distribution of cation, anions on the CEC and AEC, because they are the source for soil solution. Nutrients for soil solution. The ease of replacement of calcium from colloids varies in this order: peat, kaolinite, illite, montmorillonite. I think these issues I discussed somewhere else also. And 80% calcium saturated 2:1 clay provides the same percentage of calcium release as 35% calcium saturated kaolinite or a 25%. So try to read, try to understand what does it all mean. Ion absorption by plant. Now we go little inside. means now your nutrients are to be taken up by plants into their roots. So once nutrients are at root surface, they enter the outermost root cells where they are transported through the plant. They have to go through the, means they have to follow the radial movement. Radial movement. Ion transport out of the root also occur and can influence rhizospheric pH. So certain ions come out of the root also. They come outside the root like, hydrogen ion, hydroxyl ions. They come out of the root and they help in the exchange of ions. Thus, it is important to understand how ions and water move into plant roots. But some basics we need to know, like transport system in plants. What is transport system? So plant stem and roots transport substances around plant. Two type of transport systems are there in the plant. Transport system from root to shoot or from shoot to root. So they are xylem and phloem. It is very fundamental. Everybody of you knows it. Xylem transport water and minerals. You know it. Means minerals have to move through the xylem. Water has to move through the xylem. Phloem transports sugar and amino acids for manufactured food dissolved in water. These are distributed differently in roots and stems. Differently means it depends upon the nature of plant, kind of plant. Some may accumulate in root more, like you get root vegetables. Some may accumulate more in their leaves. Some may accumulate more in stems. So they use different. It is as per nature of the plant. In the root, the xylem forms a central column. It is central part. It forms a solid support, the xylem. So I think you need to understand or need to see the structure of the root. The phloem is towards the center, outside the xylem. You will see now. In the stem, the transport tissues of the xylem and phloem are grouped into vascular bundles. So their group is also called as vascular bundle. Therefore, sometimes, these angiospermic plants are called as vascular plants. You can see transport system in plants. So this is a section through a plant root. So plant root, different parts of the plant root you can see. So you can see outside our root here, and this is transverse section of the root. This is parallel to the ground. So, and then you'll get cortex cells and some other cells. So you get phloem here. Phloem is here in the root. Phloem is here. It is towards the central part, and xylem is in the central part. So this is your xylem part. This is hypothetical diagram you can see. And then if you see the structure of the stem, in the stem you get these kind of structures, and they are representative. These sections, the pictures, they will vary with the kind of species, whether it is monocot, dicot, or different, but it is a very general representation of xylem and phloem. So here you can see section of the plant stem. So it is a bundle. Bundle has two kind of things. One is your xylem inside. Xylem is inside this circle, and then phloem is outside. So these are all phloem, and inside this is xylem. And xylem is many times it will accumulate lignin and some other substances. So therefore it is also considered many times a dead tissue. So you know about vascular bundle, phloem and xylem. Xylem will transport water and nutrients, and phloem with carbohydrates, acids, amino acids, and so on. Xylem. The xylem transport water and minerals from root up to the plant stem into the leaves. So it distributes this xylem. Once your nutrient or water enters the xylem, it will distribute to all other parts of the plant. In a mature flowering plants, most of the cells that make up xylem are specialized cells called vessels. So there are different kind of xylem cells also. I'm not going into the details, but for transport of nutrient and water, this vessels, xylem vessels are more important. So vessels, they lose their end walls, so the xylem forms a continuous hollow tube through the plant system. And one more structure was there, the central part. The central part is also called as stele. Stele or stele, I don't know. S-T-E-L-E, stele. S-T-E-L-E, stele. The central part is your stele or pith, P-I-T-H, pith. So vessels become, lose their end walls. Vessels become strengthened by a chemical called lignin. Lignin accumulation can happen. And again, its concentration will vary with the species. Some may be annual, biennial, perennial, grasses, dicots. So accumulation of lignin will vary with the species. Cells are no longer alive. These xylem cells or vessels. Lignin gives strength and support the plant. So this xylem is very, very important. It has lignin, which gives support and strength to the plant. And we call lignified cells wood in some woody plants. Lignified cells wood. So you can see the structure of the xylem. Transport in the xylem is a physical process. Once it has entered the nutrient or water has entered the xylem vessels, then transport is a physical process. It does not require energy. So this is, I got from bbc.co.uk. I put the slide. You can know a little more on this. Now, very simple diagram suggesting the role of xylem vessels in the transport of water. This is for water, and almost similar role will be played for similar will happen in the uptake of nutrients also. Almost similar root will be almost same. So soil water, higher water potential. If this is soil or solution, and this is root here. And you should remember that nutrient uptake can happen from any part of the root. Any part of the root can take up nutrient. And there are different parts of the root. Some part are taking up a particular nutrient. The nutrient uptake will also vary with the root part. And most of the time students consider that root hair are always used for the uptake of the nutrients, but that is not true. Not just root hairs, but many other parts of the root are responsible for uptake of different ions or molecules. And so far water is concerned, water uptake mostly happens by root hairs, but other parts of the root also help in the uptake of water. So here you can see that water is through root hair, and it will enter the cortex. In the beginning, there may be epidermis and then some other layers, cortex. So it will pass through the cortex, and ultimately it has to reach the xylem. And from xylem, it will be go up or down. It will be distributed in the roots and other parts of the plants through xylem vessels. So this is the simplest diagram to understand water uptake. Now you can see this is the case of distribution of water. This is again for water, how it is transporting. This is transport of water. Uptake of water ends when it enters the vessels. And from vessels, it will be transported to other parts. So you can see water surrounding in soil. Root hair takes up the water, and then cortex cells and all kind of cells, and then ultimately it reaches the xylem column. So xylem, and then from xylem, through xylem of the root and stem, it will go to the xylem of the leaves and other part. And in leaves, if you see the veins, these veins contain xylem. Veins and smaller veins, they contain xylem, and through these xylem, the water will reach the leaf. So it is continuous. The xylem is continuous, starting from root to the leaf and any other part of the plant. So xylem contains a continuous column of water. You can see this is continuous column of water. This side also maybe petiole, maybe branch, and so on. So xylem in vein supplying the leaf this water. So this is simplified thing, and almost similar movement and uptake will happen for the water also, because for the nutrients, because nutrients will also follow the xylem for its distribution in the plant. Now see the structure of the root, very simple structure of the root. And you can see this is longitudinal section. This is LS, longitudinal section. You can see root hairs are not uniformly distributed on the roots. This is just one typical example, but this may have rootlets, and then rootlets can also have some root hairs. But root hairs are not uniformly distributed throughout the roots. In some sections, some parts, you will find root hairs. So this is your root hair zone here. And then zone of maturation. This is zone of maturation up to certain length. And then zone of elongation, this area. And then you get root cap. Very general example of a dicot root, it may be. And then here this apical cap covers the apical meristem, where the cell division happens and cells multiply and root growth happen. And then if you see outside layer, the most outside layer is epidermis. Epidermis, one layer is there. And then inside this, this may be cortex. The next layers are cortex layers. You can see this cortex. So these 4 or 5 layers after epidermis are cortex. And after cortex, then you have a layer of endodermis. Endodermis layers are there. You can see this endodermis. And then one more comes, that is pericycle. Pericycle is also there, maybe single layered or multi-layered. So here it is shown single layered, I think. It is single layered. And then you get xylem inside. Xylem is inside. This green one is phloem. This is xylem in the central part and surrounded by phloem. So this is very simple representative structure of a root, outside structure and inside structure, so that we can know that how much a nutrient is going to travel. First, it has to come to the root surface, any part or root hair, including root hair. And then from here, it will enter so many cells. It has to pass a great hurdle to reach the xylem. Now see some more close picture. This is from Britannica, Encyclopedia Britannica, this picture. You can see this root hairs are on the epidermis, the outer layer. And after epidermis is a single layer, single cell layer. You can see, and then one layer of cells. And there are many layers of cells, 1 layer, 2 layer, 3. It will vary with the species, and these are your cortex area. And then you get endodermis. One endodermis is there, and then one pericycle may be there inside the endodermis. So this is one cell layer. So endodermis, inside it is your pericycle, and then in the center it is xylem, and then surrounded by phloem. So for better understanding, you can see. So your nutrient has to start with the root hair or other part of the root. It has to pass several kind of cells or intercellular spaces. It must reach the xylem. So it is not very simple job. These plant physiologists or biochemists have done a lot of hard work to know it. And there are many theories, many means. No single answer is available. Now you see, these are the parts or areas of the roots for nutrient absorption. So remember, root hairs are not always the source of nutrient uptake. Many root parts are important. For example, in case of barley, for calcium, root apical region is important for calcium uptake or absorption. For barley, for example, for iron uptake, root apical region. For corn, iron, it is entire root surface. For several crops, potassium nitrate, ammonium phosphate, entire root surface. root elongation zone, root apical region. So entire root surface. So what is the message? You don't bother about it. You just remember one thing, that all parts of the root are involved in the uptake of ions. You cannot specify that the nutrients are taken up by this section of the root or that section of the root. The whole root, entire root surface is involved in the uptake of nutrients. However, if you want to see nutrient specific, you can see it. So many people have done these kind of research, and information is available. Now, nutrient uptake by plant roots. So nutrients come in contact with the plant roots by root interception. You know how they are coming close to the roots, mass flow or diffusion. These nutrients will now enter into the plant roots and get transported into the plant cells. And they have to pass through the cells. These cells in picture, you can see, you can remember again, epidermis, your cortex, and then your endodermis, then pericycle, then it comes your xylem cells. So it have to enter variety of cells. So nutrient uptake by plants can be addressed into two parts. One is uptake of nutrients or ion, that is acquisition by roots from the rhizosphere. Means it must enter first time into the roots. So uptake of nutrients ion from the rhizosphere. Number two, uptake and transport in the plant cells or through the plant cells system. Nutrient and water uptake happens at the root hairs and the rest of the root dermis, root epidermis. So root hair and rest of the epidermis. This point is very important, otherwise you will be in confusion. In many books, you will find that water uptake happens only by the root hairs. Get it clear by yourself. In many books, it is written that root hairs take up the nutrient also. But matter of fact is that nutrient and water uptake happens at the root hairs and the rest of the root epidermis. Means it is not exclusive right of root hair to take up water or nutrients. But most of the water is taken by root hairs, but some water can be taken by other parts of the root. Similarly, most of the nutrient uptake is by other parts of the roots, but some nutrient uptake can happen by root hair also. That is the main point I could find from literature. Now, uptake from soil solution.
Sir, please repeat, sir, once, sir.
I repeat that water uptake is mostly by root hair, but it can be taken up by other parts of the root. Like you have seen elongation zone, maturation zone, apical parts of the root, different parts of the root. So water can be taken up by other parts of the root. It is not exclusive right of the root hair, but most of the water uptake happens by root hairs. So far nutrient uptake is concerned, acquisition of nutrient is concerned, it is mostly by the root parts other than the root hair. But it is also some nutrients, some proportion of nutrient is taken up by root hair also. But most of the nutrient uptake happens through the through the parts other than the root hair.
Okay, sir.
Is it clear? And I request.
Yes, sir, clear, sir.
Yeah, I request you to search yourself and go to the final, but don't believe one author. One author or one paper should not be believed in this matter, because I have seen some books, many papers, and I found different ideas, different things, believe me. And then I tried to summarize it so that we get some meaning out of it. Otherwise, there is no clear-cut idea. You do it yourself and you will realize about it. Now, uptake from soil solution can be described by passive and active processes. So now it is simple uptake. It is just entering into the roots. So here also you get active and passive uptake. There can be active and passive transport also. So uptake from soil solution can be described by passive and active processes. Ions passively move to the boundary through which ions are actively transported to organs in plant cells. Solution composition, that is ion concentration, outside and inside of the boundary, controlled by different processes, each essential to plant nutrition and growth. You will get more clear in the next slide. Now we are just dealing with now let us deal with the absorption, and accumulation. You will understand, means these three actions are very, very important in the entry into the cell. How they are going to enter into the cells, these ions. So adsorption, you understand, they will be coming to the root surface and they will be adsorbed to the roots. Then absorption, then they will enter, and then their accumulation inside the cell wall. And then how it will move further, that is another case. It is up to reaching to the cell membrane. And then from cell membrane onward, the different process will come. So the uptake of cations begins with the adsorption of the cations to the cell walls, where polysaccharides carry negative charges. So polysaccharides are there in the cell wall. They are positively charged. Negatively charged. The uptake of cations begin with the adsorption of the cations to the cell walls. They will be attached to the cell walls where polysaccharides carry negative charge on the cell wall, which attract hydrogen ions moving out of cells by the action of proton pumps. So they will be the hydrogen ions will be attached to the polysaccharides of the cell wall. Now these hydrogen ions then accessible to be exchanged for soil solution cations, which thus become electrostatically bound to the cell wall. Means the exchange will happen with the hydrogen and cation, which is outside the cell wall. So this hydrogen will exchange for the cation, and the cation will enter into the cell wall, and then this hydrogen will come out. The binding sites lie not just on outer surface of walls, but inside the capillary spaces within the cell walls. So there are some capillary spaces within the cell wall where the binding site lies not just binding site for this polysaccharides lies not just on the outer surface of the walls, but inside the capillary spaces within the cell walls. There is also the possibility of some cation exchange, termed contact exchange, between hydrogen ions adsorbed on the root surface and the cations adsorbed onto the colloidal soil particles. Means these cations can also enter with the direct exchange. You have seen already contact exchange or root exchange. So these cations can exchange through the contact also, but other mechanism is by exchange from the soil solution. Soil solution, the nutrients are on the surface of the root, and then through exchange with hydrogen ions. So anions have few adsorption sites in cell walls. Now how the anions will move into the cell? So anions have few adsorption sites in cell walls, not very high. Few adsorption sites are there. They must simply diffuse into water-filled capillary spaces of the cell walls before passing through the plasma level. So it is really quite complicated subject. Now anions are moving into the cell wall just at their own by diffusion. The positive or cations are moving through exchange, and anions are just moving through the diffusion into the cell wall area. Or maybe there may be some positive there may be some adsorption sites which will, there may be some positive sites also in these cell walls where these negative charge will be adsorbed. To some extent, they follow the cations by electrostatic attraction. Just they follow it. Okay, they are going, let us, you also move there. So such kind of theories are happening in the nutrient uptake. I will continue tomorrow this lecture, and we will meet the same time. thank you very much, and hope now I suggest you to read from Tisdale book this subject, and I will also send you presentation. Dear students, good morning to all of you. I'm very happy to be with you on this Sunday, 11th September 2022. I am Dr. Dinesh Kumar, Principal Scientist from Agronomy Division, IERI, New Delhi. We continue with the topic movement of nutrients in soil and their uptake. We started with the objective to know the mechanism of movement of ions to plant roots. So how nutrients move to the plant roots? We know there are three ways. In two ways, nutrients are moving to the roots from the soil solution. In another means, the roots are reaching the nutrients. So that is, many people include as part of the nutrient uptake, the movement of ions from soil solution to the roots or roots reaching the nutrient or ions is also part of nutrient uptake. And then describe the process in detail, how they exactly enter into the root and how they get transported from cell wall up to the xylem. So that is, and we got some terminology in the last lecture. And then movement of nutrients from soil to roots. So root interception or contact exchange, you can say, and then mass flow and diffusion. These are the three means by which there is contact of roots and nutrients. And we discussed in detail root interception, how it happens, if you can recall. And then we have seen, for example, nitrogen is mostly moving to the roots by mass flow, and P and K mostly by diffusion. And then we have seen the role of mycorrhiza also, how they are important. They become part of roots and they increase the root absorption area. So there are multiple roles of mycorrhiza. I told you ectomycorrhiza and AM fungi. There are two categories, and for us, for field crops, or even horticultural crops in tropical, subtropical areas, countries, it is AM fungi, arbuscular mycorrhiza, which is endophyte. Kind of endophyte means larger part of this mycorrhiza are inside the roots, and they play wider role, many roles. Some roles are in the solubilization of nutrient. They release some compounds, chemicals, or organic materials into the soil that solubilize certain nutrients, and then they are made available. This is number one. Number two, these mycorrhiza can access the soil volume or soil area. They can reach to the soil areas where roots cannot reach. So they extract the nutrients from other parts of the soil. And of course, transport of nutrients from soil into the plant cells or into the roots, that is another job. And one more important job I wish to tell you is in the transfer of nitrogen from legume to cereal plant or associated plant. So research suggests that these mycorrhiza make a bridge between legume and non-legume plants, and they transfer nitrogen, and up to 30% of the nitrogen which is fixed by the legume can be transferred to the associated crop. So many times you must have seen that intercropping results in better growth of the associated crop, maybe cereal or oilseed. It is basically because of current transfer of nitrogen from the legumes. That is another interesting story. And another point is that these mycorrhiza help in the uptake of almost all the nutrients, including your phosphorus, zinc, potassium, and many other nutrients. So role of mycorrhiza was also discussed. And then we have seen mass flow. So mass flow in detail we have seen, and diffusion. So they are all part of nutrient uptake, this mass flow, diffusion, contact exchange. And ion absorption by plant. I gave you some fundamentals, some basic aspects related to ion absorption. But before that, you should know what are the different parts of the root. They are important. And for this, last night I sent you very clear and very simple information in the form of 2 chapters. They deal in detail. In the first, you will see root structure and what are the important parts of the root and what are their roles. So you must know it in the simplest language there. And you also seen the transverse section of the root and what are different parts of the root. So here I wish to tell you the role of Casparian strip is very important in this case. So you can see in the central part, you can see xylem, and on the periphery of xylem, you can see phloem. Phloem yellow color, phloem is also there. And then comes the one layer. One layer is there, which is perhaps endodermis. And this endodermis is having bands of Casparian cells. You can see small bands are there. They are the main hindrance in the transport of ions, and particularly apoplastic movement that is stopped by Casparian bands. And then we have seen xylem, and it is a continuous column in the plant. And you have also seen the major route of water and nutrient uptake, starting from root here or maybe any part of the root. Water can enter and then through cortex and different cells reaching the xylem. And you can also see how it moves in xylem. Once it is in xylems, it can go to the leaves and other part of the plant. And the prominent veins which are visible on the leaf, they have this xylem. You've seen different parts of the roots. And this one also. So you can see endodermis. So endodermis is having some bands. Bands, they are just rounding the cells of endodermis. Those are Casparian cells. And you can see different areas of roots for nutrient absorption and nutrient uptake by plant roots. So first part is uptake of nutrients ions by roots from the rhizosphere. Means the nutrients are coming in the contact of the roots, and then uptake and transport in the plant cells. So there are 2 parts. And then I started with adsorption, absorption and accumulation. Adsorption, you know, they are coming to the roots. Absorption, they are entering the root, and then they are accumulating initially in the cell wall area. And from cell wall, they will move through the plasma membrane, then they enter the cells. So this was discussed, I think. The adsorption of ions may be succeeded or may be followed by absorption, passes through the plasma membrane. Next step is passes through the plasma membrane. There is an important difference. Adsorption is attraction outside the plasma membrane, and absorption is actual entry into the cell, to the inside of the plasma membrane. That is your absorption. When the ions are taken up to a greater concentration inside the plasma membrane, then outside, this is termed accumulation. They are getting accumulated. Now you can see in this picture, uptake, then absorption, adsorption, and then absorption. And finally, they get accumulated. Now, let us see some broader picture of ion uptake and transport. I have not started exact understanding of the nutrient uptake. This is just, I am discussing background. I'm not discussing in detail the mechanism. That will come shortly. So with this background, at least we can get some idea. Ion absorption by roots is more pronounced in the root hair zone than in the meristem and elongation zone. But that does not mean that it will not happen in meristem and elongation zone. So root, any part of the root can absorb ions. The root hairs are simply extension of specific epidermal cells that greatly increase the surface area available for ion absorption. So root hairs always originate from the epidermis. So epidermal extension of certain epidermal cells are the root hairs. An ion that enters a root may immediately enter the symplast. So there are 2 pathways or 2 routes through which ion move from, say, plasma membrane up to the Casparian strips or up to the endodermis. Up to endodermis, there is not much issue. So an ion that enters a root may immediately enter the symplast. Symplast is from cell to cell by crossing the plasma membrane of an epidermal cell, or it may enter the apoplast and diffuse between the epidermal cells through the cell walls. You will understand it better when I show you this picture. So you can see the first picture here. These are routes of water and ion transport across various regions of plant roots. So you can see here, root here, epidermis, cortex tissues are there. And then there is some endodermis. Endodermis here, it is shown by pink color. And this endodermis will have certain bands or Casparian strips. So these two things are embedded. They are made for each other. Means this Casparian strip, it is overlapping the endodermis, you can say, at certain places. It is not continuous endoderm, this Casparian. And then you got xylem and phloem. And this section, particularly this section, is enlarged here. So you can see here, this is root here, and the ion has entered into the root here. And now how it can go? So it can go through two paths. One is apoplastic. Apoplastic is area between plasma lemma and cell wall. You can see this is apoplastic movement. This, and it can go through like this. You can see, I don't know whether you are able to see it or not. So this is apoplastic movement. But when it reaches the endodermis, then it cannot enter the xylem through the apoplastic movement or apoplastic route. Then it has to enter the cell up to Casparian strip or up to this endodermis. It is apoplastic movement, no problem. It will go up to here, but from this point onward, it has to follow the symplastic movement. Then the apoplastic ions will move into the cells and then they will be loaded into the xylem. This is known as loading of nutrients in xylem. And then similarly, you will get symplastic pathway. Symplastic pathway is through the cells, and you can see some structures are there, plasmodesma or plasmodesmata. And you can see these arrows, this is symplastic movement. And in the end, you can see both symplastic and apoplastic. They are just passing through the cells and reaching the xylem. Hope it is clear to you. You can see Casparian strip. It is on the endodermis. It is on the endodermis. Any doubt, any point you want to ask in this picture?
No, sir.
Okay. So this is the first time I'm dealing with this subject, and there is a lot of confusion. You will also realize no author has written this subject very clearly and cleanly. Every time you start some it is really very confusing. But even there may be few errors in my way. But I want to give you something more clearer than what is there in the books. Now, from apoplast of cortex, apoplast of cortex means the part between two cells, external part between two cells. Cortex and ion may either be transported across the plasma membrane of a cortical cell, thus entering the symplast, or diffuse radially all the way to the endodermis via the apoplast. Now I will show you what is apoplast and symplast. Apoplast forms a continuous phase from the root surface through the cortex. Cortex cells are there. However, in all cases, ion must enter the symplast before they can enter the stel. Stel is your central part where xylem is there, or you can say before it can enter the xylem, because of the presence of Casparian strip. This Casparian strip stops the apoplastic movement. Now both roots merge into one, that is symplast. And what is stel? Stel is central part of root or stem system containing the pericycle and vascular bundles, xylem and phloem. That is central part of the root or stem. The Casparian strip is suberized. Suberized means it gets somewhat thick. Suberized layer that forms rings around cells. It's forming rings or bands around the cells of the specialized endodermis. So you can see point here, and effectively blocks the entry of water and solutes. Solutes may be ions or molecules into the stele via the apoplast. Now there will not be any more this apoplast after Casparian strips. Once an ion has entered the stele through the symplastic connection across the endodermis, it continues to diffuse from cell to cell into the xylem. Then there will not be any issue once it is crossing the endodermis. Once ions have been taken up into the symplast of the root at the epidermis or cortex, they must be loaded into tracheids or vessel element. Means they are part of xylem, this tracheids and vessels of the stele to be translocated to the shoot. So all ions have to reach the xylem. The presence of Casparian strip allows the plant to maintain a higher ion concentration in the xylem. That is the advantage. Means it is, on the one hand, it is stopping the entry through apoplast, but on the other hand, it is not allowing the absorbed ion or transported ion which are present in xylem back to the cortex. So that is the advantage. Plant to maintain a higher ion concentration in the xylem than exists in the soil water surrounding the roots or other root cells. Now this is the point, we can summarize the ion uptake. So mechanism of nutrient uptake and transport in the plant cell. So this subject is always together. Mechanism of uptake and transport. Uptake will be completed only when it is reaching the xylem. Now you see nutrient uptake by roots. So this is, I have taken from Karthika 2018. She published very good information on this nutrient uptake. So nutrient uptake by roots, apoplast, transmembrane, symplast pathway. Nutrient uptake. There are three important aspects in this nutrient uptake by roots: apoplast, transmembrane, and symplast pathway. So at first, the nutrient will be deciding, or nutrient will be going to apoplast. It can go to apoplast. It can go to transmembrane. That is your lemma, plasma lemma, and symplast pathways. Enter the cells. Then uptake of ions by diffusion or by cation exchange. This is already discussed in movement of nutrient to the plant root, how they move to the plant root. So that is also part of nutrient uptake by roots. This is nutrient uptake by roots. So uptake of ions is by two means. One is diffusion by cation exchange. Then comes the transport or movement of nutrients or ions into plant cells. How they move. Then passive transport. One is passive transport. So in passive transport, ion exchange theory. This is ion exchange theory or contact exchange. And then Donan effect and equilibrium. Donan effect and equilibrium. And then mass flow. So this is passive transport. You can see up to this ions are just entering the root up to this. Then active transport. Active transport is active ATP mediated ATPase pumps. So one option is ATP mediated ATPase pumps. Second is cytochrome pumps, and third is phospholipid as carrier. So these are the three theories, you can say theories or means or ways of active transport. But in all the three, it is called active because of involvement of energy. Dear students, I am not very much convinced or do not have very much high idea about this, because this is really not in my domain. I did not study this subject in detail. This is area of plant physiology, basically, you know it. So if you are attempting any course in plant physiology, you can know it better. But anyhow, I am doing my best whatever is possible. So active transport involving energy is through ATP mediated ATPase pumps, then cytochrome pumps, and then phospholipid as carrier. So this is your active transport. Then comes the membrane transport. Membrane transport is by three different means, through channels, how they can pass through the membranes. So through channels, through pumps, or through carriers. So there are three ways by which membrane transport can happen: channels, pumps, and carrier. These pumps can be further divided into two for membrane transport: primary active transport. Here, primary active transport, wherever active is there, energy is involved, and energy will come from ATP. Then in pumps, you got secondary active transport. Secondary active transport, primary active transport. In secondary, you get two. One is Simplas, uh, co-transport. Co-transport is the other word for Simplas, and antiport. Antiport is counter transport. So this is how this completes the mechanism of nutrient uptake and transport in plant cells. So hope this chart will help us in getting better understanding. Now I can continue further. Now first we go to apoplast, transmembrane, and Simplast pathways. So these are pathways, and nobody is sure. Everybody has proposed, so many theories are there. So many theories are there. That is why we don't have one solid answer for nutrient uptake and transport. So let us see how apoplast, transmembrane, and Simplast pathway happen. This is part here. You can see nutrient uptake by root, apoplast, transmembrane, and Simplast pathway. So free intercellular spaces means some spaces are available between two cells. Free intercellular spaces of epidermal and cortical cells are called apoplast. Okay, you got the meaning of this apoplast. Free intercellular spaces of these spaces are in epidermal cell as well as in cortical cells. So these are your apoplast. What they are doing, it facilitates movement of ions or water in root and does not need movement through impermeable membrane. So here impermeable membrane is not there. They can do their job through this route. Then Casparian strips force water and ions in the apoplastic region to cross the endodermis by passing through the plasma membrane. Casparian strips force water or ions in the apoplastic region to cross the endodermis by passing through the plasma membrane. The cellular connections and plasmodesma help in movement of ions from cell to cell in symplastic movement. So in symplastic movement, you get plasmodesma. Plasmodesma help in the cell-to-cell transfer of ions. You can see this picture very clearly shows you apoplast and symplast. So here you get some intercellular spaces. You get cell wall and cell membrane. And there are some interspaces. And then, for example, this is apoplast. So it will enter here and it will go to the next apoplast of the next cell. And it will go like this. So this is your apoplastic movement, and symplastic is lighter color. You can see from this cell to this cell. So you get these connections through plasmodesma. Plasmodesma is singular and plasmodesmata is plural. Plasmodesmata plural and plasmodesma singular. So from this cell to this cell, they have the way through plasmodesmata or desma. So you can see apoplast pathway through cell. This is this part, and then symplast. Symplast is from cell to cell. And sometimes they have to cross the vacuole also. Vacuole also, and this is cytoplasm. So intercellular transport of solutes through plasmodesmata is thus called symplastic movement or transport from one cell to another cell through plasmodesmata. So plasmodesmata are tubular extension of plasma membrane. Their size is 40 to 50 nanometer diameter. They traverse the cell wall and connect cytoplasm of adjacent cell. So it is connecting two cells. Cytoplasm form a continuum interconnected by it is referred as symplast. Hope it is clear to you. Now we go to the uptake of ions. Uptake of ions, as you know, it is by three different means: contact exchange, diffusion, and third one is active, that is mass flow. So it says that this classification suggests that contact exchange and your diffusion, they are kind of passive movement, passive entry, and then through mass flow, it is active entry. I have shown you here. So you can see uptake of ions by diffusion and cation exchange. This is very passive kind of uptake. This is not, this is uptake, and here again passive transport, ion exchange theory, domain effect, and mass flow. Some confusion is still there. It is your job to clear confusion. My job is to get some interest in you to read this topic in detail. So now we go to the uptake of nutrient ions. Nutrients or ions move towards plant root by diffusion or cation exchange. Diffusion occurs on the basis of concentrated gradient. We already discussed it. Cation exchange happens in response to the release of hydrogen ions. You remember this oscillation, dancing of hydrogen and potassium, causing a change in soil pH around roots. When water or ions enter plant root, they move through apoplast, transmembrane, or symplastic pathways. Afterward, movement of nutrients occurs via xylem, and nutrient uptake differs with area section of the roots. This is important. Now, transport or movement of nutrients or ions into plant cells. Plasma membrane and tonoplast. Tonoplast is the layer or membrane around the vacuole. Plasma membrane and tonoplast are the major barriers for movement of ions or nutrients and selectively regulate the movement into and outside the cell. We need to know the characteristics of these barriers, plasma membrane and tonoplast. Number one, permeable to oxygen, carbon dioxide, as well as certain compounds, but semi-permeable to water, selectively permeable to inorganic ions and organic compounds like amino acids and sugars. So it is selectively permeable, means it can allow passage of certain ions or molecules or acids, but it does not allow certain others. And nutrient ions can be transported across these barriers actively or passively. Or I can summarize again where we started. Transport or movement of nutrients, you can see there are passive transport, ion exchange theory, Donan effect, and equilibrium, and then mass flow. So in this movement through, you can see this again. This one. We are dealing with this passive transport. You can see here passive transport, active transport, and membrane transport. So here you can see passive transport. This is membrane transport. Passive transport, active transport, and membrane transport. So you can see in membrane transport, channels, pumps, primary active transport, secondary active transport, symport, antiport, and carriers. So now let us see passive transport. It occurs along the concentration gradient. Nutrients diffuse into the cell if the concentration of a nutrient ion falls less inside the cell. Energy is not required. Nutrient concentration is maintained for enhancing the nutrient absorption. So passive transport happens by three different mechanisms: ion exchange, Donan effect, and equilibrium. And third one is mass flow. All are passive transport. Ion exchange theory exchange of cations present on the soil clay, and protons. I think I discussed this contact exchange or ion exchange already. Then comes the in the passive transport, Donan effect and equilibrium. So cell membrane has a negative charge that allows anions to enter it. Plants can accumulate more than or approximately 30 times the cations passively against the concentration gradient to balance the electrochemical potential difference thus generated. Now, what is Donan equilibrium? The equilibrium defined by F.G. Donan is the ratio of positively charged ions inside to the outside will be equal to the negatively charged ion inside to the outside. So you can see that what is Donan equilibrium and its significance. Then mass flow due to active transpiration, ions take up from root to xylem to shoot xylem. Due to active transpiration, ions are taken up from root to shoot xylem. This leads to a concentration gradient and a water pressure gradient, which drives the mass flow. Then active transport happens against the concentration gradient. This type of movement involves energy. And there are three ways. Number one is ATP mediated ATPase pumps. Ions are actively absorbed by using energy released by hydrolysis of ATP. H-ion ATPase are pumps. Then cytochrome pumps. Whenever a tissue is placed in a salt solution, the associated respiratory increase is called salt respiration, which is mediated by iron-containing membrane proteins called cytochromes. Protons and electrons are produced due to dehydrogenation reaction in the inner side of the impermeable membrane due to difference in oxygen concentration. The ferric ions on outside the membrane-bound cytochrome pumps accept an electron and get reduced, and subsequently electron is transferred or carried outside by cytochrome chain. This cycle repeats after the next anion is taken up by ferric ion from outside. This concept was proposed by Lunde-Gaard in 1954. Phospholipid as carriers, acyl glyceride like lecithins. Then membrane transport can happen through channels. Channels, these are transmembrane proteins that exist as membrane pores, which selectively permit solutes based on its biochemical and biophysical properties. Entry of ions is determined by layer of hydrogen aquaporins, for example. Then membrane transport with the pumps. These are transmembrane proteins that transport ion actively by TP hydrolysis. Protons are pumped against electrochemical gradient, which builds up positive charge due to protons accumulated. There are two types of active transport. One is primary active transport. Energy from ATP hydrolysis is directly used here. And then secondary active transport due to the electrochemical potential gradient that is generated as a result of primary active transport. The protons pumps outside against the concentration gradient result in chemical potential difference. Then secondary active transport is divided into two, symport and antiport. So then third one is carriers. This concept defines a specific carrier for each ion. The enzyme phosphokinase mediates the formation of an activated carrier. So you can go through it. It was just some exposure to you, this topic. I really did not have sufficient knowledge for this subject. Therefore, I could not do the justice with this subject. But I think I was successful in at least exposing you to this topic. Now, it is a challenge for you to understand it in a better way. Of course, I did my best to send you the best information available on this subject, the best and simple information. When you start reading yourself this subject, you will also feel a lot of confusion. So next time, if I teach in the next year, then I will surely meet some plant physiologists and get the fundamentals clear. I really did not find much time to read more and more and learn and teach to you. So this ends here, and then we will go to the next link for fresh topic. So thank you for this part. We go to link 2.
Okay, sir.
Thank you, sir.